Hey guys, so last time we were talking about the evolution and diversity of plants, and today we're going to start off with chapter 17, which is the evolution and diversity of animals. So here we go. The last chapter, chapter 16, was just one video. This one is going to be 4. It is a really long chapter, and so I'm just going to break it up for y'all. That is why we are only covering chapter 16 and 17 this week. Next week, we will cover chapter 19, chapter 18, and chapter 20. Okay, 18, 19, and 20. So your evolution and diversity of animals, hold on, let me move this for a second. So animals are diverse, yet they all have features in common. Just like all living things have features in common, all animals, no matter if they are an owl or a scorpion, are going to have things in common. They've got multicellular bodies, eukaryotic cells that lack cell walls, heterotrophic by ingestion. So heterotrophs consume other organisms for their energy source. So they cannot make their own energy. They have to consume it by consuming another organism. Then they have a blastula, a hollow ball stage of development. So basically you have a pool of cells, and then that pool of cells is going to sort of start separating and forming your different cell layers. Okay. So vertebrates, there are about 57,000 known species versus invertebrates, which are a lot of, things like scorpions, things with these exoskeletons, things like insects. There are over 1 million known species of invertebrates. So when we think about animals, we tend to think of dogs, lions, tigers, bears, things like that. But I'm just pointing out to you the disparity in the actual number of species, okay? 57,000 versus over 1 million known species. So animal life began in the water, where you had, oh, hold on, that's on a different slide. But so the first animals arose from aquatic protists about 570 million years ago. You had these single-celled aquatic protists that looked a whole lot like just one of these, okay? And then all of a sudden you have multicellularity. And then all of a sudden you have tissues. And then all of a sudden you have germ layers. And as you keep going with all of those different characteristics, you're getting new types of animals. Okay, so the origin of animals in the ocean happened 570 million years ago. And then you can just go and look around. You got your first fish, your first invertebrates, first invertebrates on land, first vertebrates on land, first reptiles, mammals, primates, Homo sapiens 200,000 years ago. So most of the animal phyla that we're going to talk about are, and remember, so a phylum is a larger group of animals and organisms than we have talked about yet. So it's just like a large grouping. So if you remember, Dear King Philip came over for good spaghetti. Philip, the P, is for phylum. So many phyla will make up a kingdom, et cetera, et cetera, et cetera. We talked about that a few chapters ago, but most of the animal phyla came around during the Cambrian explosion between 530 to 570 million years ago. What? All right, so you have animal features that reflect shared ancestry, and they are characterized into 9 major phyla, which we will talk about in this chapter. Your first branching point, and what I want to bring up before we start talking about your different branching points is, and the characteristics that are included in those, is that you had this ancestral protist that is the common ancestor to all of these different animal phyla, okay? And here are all of your 9 animal phyla that we're going to be talking about: chordates, echinoderms, arthropods, roundworms, annelids, mollusks, flatworms, cnidarians, sponges. The common ancestor for all of those is in a single-celled ancestral protist. Then Over the course of time, you get multicellularity. So instead of having this single-celled organism, you have a multicelled organism, and your first branching point after multicellularity is going to distinguish organisms based on if they have no tissues versus if they have true tissues. Right there. So in sponges, so you can see right here, sponges, if you trace this back, the commonality between all sponges is that they have no tissues. They do not have true tissues. So for instance, what are different tissue types? You can have nervous tissue, digestive tissue, whatever, cardiac tissue. They do not have specific like groups of cells functioning together to perform specific tasks that you have in tissue types. All right. The second branching point distinguishes different symmetry, bilateral symmetry versus radial symmetry. And also, oh, well, hold on. All right, bilateral symmetry versus radial symmetry. And we're going to talk about those, but it also, at these same branching points, you also have organisms that are 3 germ layers versus 2 germ layers. So let's talk about radial versus bilateral symmetry. Radial symmetry is where you have a central axis to that organism. Okay, so for instance, I think that this is a hydra, or if you pictured like a starfish, you can make a cut anywhere along that central axis and you'll get a mirror image on each side, no matter where you cut it along that central axis. That is radial symmetry, okay? Mirror image on either side, no matter where you cut it. Versus bilateral symmetry, this is where you only have one plane where you can cut this organism in half that would be mirror images on both sides. So that's bilateral symmetry versus radial symmetry. And what's awesome, I said radial symmetry has more of this central axis. So it has the central part of the organism and then branch off points from the middle. Versus in bilateral symmetry, you have a lot of cephalization that is typical, meaning that cephalization, you have a head and you have organs located in that head, okay? So if you have a distinct head, it's more likely that you're gonna have bilateral symmetry. The same branching point also distinguishes animals with 2 or 3 embryonic germ layers. So basically how we get to the different germ layers is first, you have this ball of cells, this embryonic ball of cells called a blastula. It is basically a hollow ball of cells, okay? Then this hollow ball of cells starts folding in on itself. And when it starts folding in on itself, it moves from being a blastula to being a gastrula, okay? And this gastrula, if it only has these inner cell layers called the endoderm and this outer cell layer called the ectoderm, then it's got 2 tissue layers. Versus if it has the same ectoderm and the same endoderm, but also has a 3rd cell layer called the mesoderm, then it's got 3 tissue layers. So that's the difference between 2 and 3 cell layers in your gastrula. 2 cell layers, you just have that endoderm and ectoderm. In the 3 tissue layers, you have an endoderm, ectoderm, and a mesoderm that develops. And they're going to give rise to different body parts in these organisms. All right, so here is a life cycle of a starfish. Okay, so starfish, adult starfish goes through meiosis. You make an egg. That egg is fertilized by a sperm. You have a zygote. That zygote is going to start dividing until you have the blastula, that hollow ball of cells. That blastula will start sort of folding in on itself. Now it's an early gastrula. Later gastrula, you have distinct cell layers. So this is your ectoderm out here. And in here, on the part that has folded in, okay, this is going to make your endoderm. And your endoderm, this inner cell layer right here, is going to make up your primitive gut. So as the gastrula moves on from just, different cell layers to being an actual starfish larva, you're going to have totally different tissue types. And that endoderm now forms your digestive tract, okay? All right, so germ layers of the gastrula. One, you have your ectoderm, which is gonna form your epidermis and your nervous system. Then for this starfish does not have a mesoderm, okay? Does not have a mesoderm, so let's overlook this for a second. And then the endoderm is gonna form this yellow digestive tract. If it did have a mesoderm, the mesoderm would form lots of other internal organs. So for instance, your mesoderm, this middle cell layer, would form cardiac muscle, skeletal muscle cells, kidney cells, blood cells, smooth muscle in the gut, for instance. So the mesoderm is a lot of these other things. Okay, so another branching point, the third branching point, is a lot about embryonic development. So in that gastrula, okay, that indentation, is the mouth forming first or is the anus forming first? And that is going to be a huge part of embryonic development and another branching point. So if you have this, if you have this gastrula that is, you have this indentation, and like we said before, this endoderm is going to become your digestive system. If this indentation becomes the mouth, that first indentation becomes the mouth, you are a protostome, okay? If that first indentation becomes the anus, if the anus is formed first, you are a deuterostome, okay? So here it is again. If that indentation becomes the mouth, you're a protostome. If it becomes the anus, you're a deuterostome. Now, if you are a deuterostome, what will happen is that you will form the mouth once, like, this will come and sort of connect with this side. So it's all, you will form the mouth on this side and anus will be on this side. So, oh my God, Cooper, I'm pulling on your face, get down. Okay, get down. You will form the mouth afterwards, okay? Yeah. So if it develops into the anus, the organism is a deuterostome. What was here? Other things that characterize animals are going to be the body cavity, the organization of the digestive tract, and segmentation. So let's talk about those real quick. Koopy. Okay, so in bilaterally symmetrical animals, so we are bilateral symmetry, all of these organisms, from flatworms, mollusks, annelids, roundworms, arthropods, echinoderms, and chordates, all of these have bilateral symmetry. Most of them have this cephalization, meaning that they've got a head. An obvious exception to that are the echinoderms. They've got 3 cell layers. And in these bilaterally symmetrical animals, where you have these 3 germ layers, right? Because here, bilateral symmetry, and you have the 3 germ layers, you might have this body cavity. So blue is your ectoderm, okay? Blue is your ectoderm. It's gonna make like your skin and your nervous system, versus your mesoderm here in pink. And then in yellow right here, your endoderm, that's gonna form your gut. So if you have a column is just a body cavity, okay? So here, in this sheep, for instance, you have a full column, a full body cavity. It is completely surrounded by mesoderm, versus in a pseudocolumn, it is not completely surrounded by mesoderm. Here, it is surrounded partially by mesoderm, here in the pink, and partially by endoderm. So that's a pseudocolumn. And then you can also have no column at all, where you just have ectoderm, mesoderm, endoderm. So no body cavity. Column, fluid-filled cavity that forms completely within the mesoderm. That is right here. So it's only touching mesoderm. then a pseudocolum, partially lined with mesoderm here in pink, and partially lined with endoderm in yellow, that's the gut. And then no column whatsoever, you just have those 3 germ layers. So animals, we're also talking about digestive tracts. So an animal can have an incomplete digestive tract, and in an incomplete digestive tract, what's gonna happen is that this organism will take in food into its digestive tract. It's got its gastrovascular cavity. It will digest the food, absorb the nutrients, and then eliminate the waste out of the same hole before it can eat again. Okay, so that's an incomplete digestive tract. The mouth takes in food and ejects waste. Then you have complete digestive tracts where organisms like us can take in food, digest that food, absorb the nutrients from that food, and eliminate waste, but through different holes, right? So that means that once you eat, it's going through the stuff that it's going to go through. You're not regurgitating your waste out of your mouth. It is going out through your anus. So before you have eliminated your previous food, you're able to eat again. Now we're talking about segmented bodies. Some animals have these segmented bodies. You can see the segmentation, like the millipede and the centipede. This is awesome for flexibility, makes a hugely flexible organism, and also potential for developing specialized body parts, more so than unsegmented bodies. All right, guys, different combinations of all of these qualities that we have talked about are going to characterize each of these phyla, just like when we were talking about your different groups of plants, okay? So we are going to stop here, and in the next video, we are going to start talking about some of these phyla and how they differ. All these different combinations are going to lead to different phyla. Okay? Okay, I'm going to stop share. Guys, hope you're excited about animal diversity, and I will see you next time.